But today we're going to finish up talking about HIV, as well as finish our conversation about vaccines. And in fact, I feel like most of what we have to say kind of fits together pretty nicely with this material. So we talked last time about HIV and all the reasons why AIDS is bad. And what I want to talk about today is sort of ways that we can actually intervene to deal with HIV. One of the things that we can do is to use medications in people who are already infected to improve their condition with their HIV infection and with AIDS. This involves the use of some drugs called anti-retroviral therapies. And one way that you can sort of divide a lot of the things that I'm talking about today is between things that we do for a patient when they are already infected to try to make them feel better versus things that we do to prevent infection. So these things that we're talking about with anti-retroviral therapies are what are known as therapeutic interventions. They are done after infection as opposed to prophylactic interventions, which are done to prevent infection. Anti-retroviral therapy has been revolutionary in our view of the HIV epidemic. So in red, you can see the deaths per year due to AIDS. And in blue, you can see the use of one type of therapy for AIDS. And you can see that as that therapy's use increased, the number of people who died of HIV decreased dramatically. And so for most people with HIV, these therapies are allowing them to live with their infection for a much longer time. This therapy, in a big picture term, works by reducing the level of viral load. So at the beginning, people will have high levels of virus in their blood, and once we start taking antiretroviral therapy, that level of virus in their blood will decrease until we get to the limit of detection of the virus in our assays. And so oftentimes these patients will be referred to as undetectable because their virus is under control and cannot be detected with our typical assays. We have developed more and more sensitive assays to measure the virus, and we often will find some patients who are detectable and some who are even lower than that detection limit. However, one thing to realize with these antiretroviral therapies is that it does not actually reduce the amount of virus to zero. Virus does not completely go away. It just goes to a level that is below that limit of detection. So the virus is still there in the patient's body. There's just a very small amount of it that's hard to detect, but it is still present. Based on the information that we have, it is relatively easy to understand in somewhat more detail how these antiretroviral therapies work. Basically, each of the drugs that exists as some type of antiretroviral therapy inhibits one of the parts of the replication cycle that I presented to you last time. The first set of drugs that were described were the nucleoside analogs. These work by having a structure that is very similar to the structure of one of the nucleotide bases in DNA. The most famous of them is AZT. AZT looks a lot like thymine or thymidine. It's actually what the T is for. But instead of having a hydroxyl group at the end, at the 3' end, where you could add on new bases, AZT has an azide group, AZ for azide. And so no new base pairs can be added on after the AZT is incorporated. And that stops the reverse transcription process. Other drugs that could bind reverse transcriptase and stop the reverse transcription process were described later. Drugs that specifically blocked HIV's protease that cleaves these precursor proteins into smaller proteins have also been described. More recently, some specific drugs that inhibit fusion, so they actually block the process of the HIV membrane, the HIV envelope, from fusing with the cell. Drugs that actually structurally mimic a ligand for CCR5 and get in between CCR5 and the virus have been discovered, as have drugs that inhibit integrase. And so each of the sort of unique steps of the virus's replication cycle can be hit by one of these anti-retroviral drug targets. And these can do a great job in stopping replication of the virus. Sometimes you will hear people talk about the fact that these drugs turn HIV into almost a manageable condition like diabetes. And you might hear people say, oh, well, because of these drugs, HIV is actually not really a big deal anymore. No one should really spend much time thinking about it. So I also want to tell you that there are some problems with these drugs. The next time someone says to you HIV is not a big deal, because they cured that, didn't they? I want to tell you some things about why these drugs are less than ideal. So first of all, in general, a regimen of one of these drugs is going to cost about $10,000 to $15,000 a year, and that is actually on the low end now. Some of the newer drugs, the ones that were described in like 2007, the integrase or chemokine inhibitors, are considerably more expensive than that. This table also shows you two other issues with the drugs. One of them is the dosing regimen, which you can see here. The times and doses of these drugs are listed. And so what you can imagine is if you had to take AZT, you would be taking 1 pill twice a day. It doesn't seem terrible. If you were taking Delaverdine, you'd be taking 4 pills 3 times a day. They must be mixed into water, and you can't take them within an hour of antacids or didanosine, which is another drug. Typically, HIV drugs are taken in cocktails together for reasons that I'll tell you about in the next slide. If you're taking Indinavir, 2 pills 3 times a day on an empty stomach or with a low-fat snack, but not within 2 hours of didanosine. And so you can see that this is a very complex regime that patients have to follow, that can be very difficult for patients to comply with. You can also see the list of side effects of these drugs on the right-hand side of this slide. You can see that some of these drugs have side effects like nausea, headache, anemia, reduced levels of neutrophils, weakness, insomnia. We also can see things like rash, headache, hepatitis, and we can get to things like kidney stones, blurred vision, metallic taste in the mouth. In many cases, abnormal distribution of fat and elevated triglyceride levels are seen, glucose intolerance, etc. And the problem with all of these things is that ideally what we want to do with these drugs is we want our patients to start taking these medications as soon as possible after diagnosis. We want to preserve their CD4 cells and their immune system for as long a time as possible by keeping the virus at a low level. This phase of time where the patients have not yet progressed to AIDS is that time that we think of as the asymptomatic phase. By and large, during this time, our patients feel just fine. And so we are saying to these patients, hey patient, you know how you feel totally fine right now? I would like you to pay $15,000 a year to follow this really difficult regimen and have all these side effects. Okay, cool. And you can imagine how people might have either financial issues with sticking with this, sort of logistical issues, or issues with the side effects in terms of sticking with this that make this very challenging. One thing that I forgot to mention on the previous slides is that there is this new movement called U=U throughout the field. We talked a little bit about the relationship between transmission and viral load levels last time. And it has been talked about in the field that when a patient has undetectable viral loads, that they are unable to transmit the virus to others. That's been talked about in the field for a long period of time. It is now at the point where even the CDC and the World Health Organization admit it and say that if you are undetectable, you can't transmit. It's now called undetectable equals untransmittable. Not only have these medications helped patients in terms of the number of HIV-related deaths, as I showed you on the first slide, we've also been able to dramatically decrease transmission because of this viral load reduction. I forgot to mention that before. But I was mentioning the problem with ARTs. There's one additional big issue with ARTs, and that is the latent reservoir. As we discussed last time, the latent reservoir involves cells that are living long term, that may undergo replication of themselves, that are only rarely going to do some viral replication and are really just going to persist. And we talked about how this gives the virus a lot of chances for mutation and allows for that escape from immune control. Well, all of the steps that the anti-retroviral therapies block that I've told you about are steps that are really important parts of the viral replication cycle. Because these latently infected cells are making very little virus, they are not susceptible to these ART drugs. The drugs are not really going to do much to them because the drugs are hitting virus replication cycle processes, and these cells are doing very little virus replication. And so these cells are not going to be hit by the antiretroviral therapy the way that the cells at the top that are actively producing virus would be. As a result, this also gives the virus a lot of opportunity to mutate. As we talked about, there is this rapid mutation that allows the virus to escape immune control. Well, here we are introducing another type of selective pressure, and we can allow the virus to mutate around that selective pressure as well. So the other problem with antiretroviral therapy that we see is that whenever a patient goes on to some antiviral therapy, like is shown here, we will get the virus under control for some period of time, but eventually the virus will in fact come back to replicate at high levels. So CD4 cells will increase for a while, but then they'll go back down. And this is because the virus will eventually acquire a mutation that allows for resistance to that drug. This is why we give our HIV patients cocktails of drugs, usually a cocktail of 3 drugs, because this puts the virus under 3 different kinds of selective pressure at the same time and lowers replication, and it slows down this mutation rate. It's harder for the virus to try to mutate in 3 directions at once while also having a lower replication rate. And patients frequently end up switching out the drugs in their cocktails in order to keep their viral progression under control. So this mutation issue is a huge problem. Any patient who has their HIV under control will eventually lose that control because the virus will mutate around control. This is also a big reason why taking antiretroviral therapies consistently is very important. There was an idea for a while that perhaps what a patient could do is take antiretroviral therapies to reduce their viral load to undetectable and then go off them for a while and then take it again and then go off it for a while. People thought that was going to be really great in terms of reducing side effects. Instead, what was seen was that people went on antiretroviral therapy, the virus decreased in number, but also had this pretty massive selective pressure put on it, so that there was massive selection for mutation. So then most of the virus became the mutant form. And then you took away the drug and let the virus replicate out of control and make huge amounts of the mutant form. And then when you went back on the drugs couldn't stop it anymore. This was originally called a structured treatment interruption. It was a bad idea. But this also points out the fact that for our patients who want to have virus under control, maintaining the drug regimen is really, really important. And that means that patient needs to be able to consistently afford these medications. They can't go on and off them. They need to be able to consistently follow these complex routines. They can't miss doses. They need to be able to deal with these side effects and not skip the drugs because of the side effects. And so this mutation problem makes a lot of the other problems worse because we cannot have people going on and off drugs. Consistency in taking this drug every day for the rest of your life is extremely important in allowing it to keep the virus under control. There are a few other types of therapies that people are investigating right now to try to improve HIV therapy that we have. One of them has to deal with T-cell exhaustion. So one problem that we know about is that in an HIV-infected person, CD8 positive T-cells will get exhausted from consistent chronic antigen exposure. And so those CD8 positive T-cells are going to be unhealthy. Perhaps even if we could get the virus pretty well under control, maybe in a perfect world, the CD8 T-cells could deliver the final knockout punch. But in reality, they can't because they're exhausted and they're in bad shape. And so people have thought about adding therapies that can reduce T-cell exhaustion to try to improve this. We actually know that exhausted T-cells have an upregulation of the specific co-stimulatory molecule, PD-1, which is bound by its ligand, PDL1, that induces additional exhaustion on that cell. And so there are drugs, pembrolizumab, nivolumab, azilizumab, avalab, I can't pronounce them. The most famous of them are Keytruda and Opdivo, that are all monoclonal antibodies that either bind PD-1 or its ligand, PDL1, to stop this interaction between them and to keep this cell from being exhausted and allow it to become an active CD8 cell or to potentially reverse this exhaustion. So the idea of treating patients with these medications to reverse exhaustion is one that people are working quite a bit on. These drugs are approved for other uses, and we are seeing them being given to some HIV patients for this use. Yeah, Molly. The cell may eventually die anyway, but particularly, as I'll get into in the sort of next couple slides, if we can do some other things to start to really push the virus down, then what we would like is we'd like the CD8 T cells to be able to fully contribute to getting rid of virus. And so oftentimes the idea is that this is going to be combined with something else. Right now, it's often combined with ARTs just to keep the viral load low, but it's hypothesized that it would be combined with some of the other things people are talking about to help make them even better as well. As I talked about, the other really big problem with HIV therapy is the latent reservoir. We can basically get rid of HIV in patients except for the latent reservoir, which is where that background mutation happens that allows the virus to get around whatever we throw at it. So if we had a way to get rid of the latent reservoir, then everything would be great because we can get rid of everything else in terms of HIV. There are two major strategies that people are interested in using in order to deal with the latent reservoir. The two of them are actually shown as the two separate arrows at the bottom of this slide. So you can see the latent reservoir is formed at the top. The two strategies are at the bottom. I'm going to talk about the one that's on the right first. And I'm going to laugh a little bit because whenever I explain this one, I think back to a class where I explained it once and did a motion and something funny happened. And I'm especially finding this funny because Vineet was in that class as a first-year student. So Vineet, this one's not going to be nearly as dramatic as last time you saw it. So the first way that people have talked about dealing with the latent reservoir is by something called shock and kill. And the idea with shock and kill is to use some type of drug to take those latent reservoir cells and kick them back. My shoe didn't fall off today, and fly across the room and hit the wall like it did the last time. Kick those cells back from their latency into being virus-producing cells. So if we can stimulate all those cells back to the idea of producing virus, we can then potentially kill them either by using ARTs to now block those virus-producing cells, or potentially by using activated CD8 T cells in order to kill those virus-producing cells. And so either way, we could make this work. So the idea is you shock the cells back out of latency and then kill them once they have emerged. You can also see this at the bottom here. The idea is that we shock the cell to actively produce virus, and then we'll kill that cell either with ARTs or with CD8 T cells. There are some really great drugs that seem to work for reversing latency. One of them that has gone quite far in clinical trials is called vorinostat. Vorinostat is an HDAC inhibitor, a histone deacetylase inhibitor that alters chromatin issues, chromatin modifications. And it's an area that I have become recently rather interested in terms of how exactly that might work. There are others that might be other types of T cell stimulators like TLR agonists or something like that that will basically activate the T cell. And so this is one of the ways people have been thinking a lot about dealing with the latent reservoir. And I will just point out for Dr. Consummate, your drug of interest, it's right there. Okay. The other big way that people think about dealing with the latent reservoir is to somehow selectively deplete the cells that have integrated HIV, to somehow target those cells and specifically kill them. And that seems like a pretty good idea, except you'd have to have a specific target on the cells. You'd have to be able to look at the cell and be like, you're the bad one, and kill it. And we'd have to have a way to know that a cell is part of the latent reservoir to actively kill that cell. That has been a major question in the field for quite a long time. And so the idea would be, well, you just figure out which cells are the latent reservoir cells. See this one. And then you just kill it, and then you're good. Hooray! For a long time, that was about as much as we could say about this theory. In early 2017, the first paper describing a potential marker for latent reservoir cells was published. This marker is CD32A. CD32A is actually an FC receptor. Why latent reservoir cells would have an FC receptor makes little to no sense. No one has really figured out why. No one has figured out if HIV likes to be latent in these cells, or if when HIV is latent in these cells, it turns on CD32, so there are lots of open questions. But the idea behind this is that you would, in theory, want to kill the CD32 positive cells. One bad piece of that is CD32 is on non-HIV infected cells too. It has another biologic role. So you'd have to have kind of a co-receptor or something to label the HIV positive CD32s instead of, like, I don't know, all the B cells with an FC receptor. You can't just kill all the CD32A positive cells. There also, within the past few months, have been some papers questioning this data that have been published in Nature. So I will say that this whole idea of CD32A marking the latent reservoir currently has a little bit of a question mark next to it in the field. But this would be one of the ideas about how we could deal with the latent reservoir. These issues are all ways that we might be able to deal with HIV in the future. There are a couple of other things I want to mention about how we deal with HIV today as well. These antiretroviral drugs that I have mentioned are outstanding in their ability to reduce mother-to-child transmission. So about 35%, we'll say, of HIV-positive moms will transmit to their babies either in utero, during delivery, or via breastfeeding. And we have a specific regimen of antiretroviral treatments that can be given to the mom in 2 different times, basically once at a certain week before delivery, once at delivery, and then mom doesn't breastfeed. And that really stops transmission of HIV to babies. And so we largely are not seeing very many kids born with HIV as we used to. All this is great in the United States. Things get much more complex in other parts of the world where it is not as easy for mom to not breastfeed because perhaps she does not have potable water in order to make formula, or perhaps there are other issues with getting formula. In those countries, there is actually a different ART regimen that is used. That's also because mom does not always go into the doctor as frequently to get those pre-delivery ART shots. And so there is a different type of regimen that is used in some other countries that also pretty severely limits the number of HIV-positive babies that are born to HIV-positive mothers. And I partially point that out because the first time I went to South Africa, Marina's laughing because she witnessed this. I was appalled at some of the things that they were doing. And then I learned there's a whole different protocol there compared to here. And I was like, you're doing what to those mothers and babies? And then I was like, oh, sorry. So yes, there is a different protocol. It is quite effective. But this sort of idea of being able to give medication to stop mother-to-child transmission also helped scientists think about using these medications in other ways. So the trick with this is that we know mom is HIV infected, and we know that she's going to transmit to the baby, and we kind of know all the details of that. If there are situations where lab workers are working with the virus, and they accidentally infect themselves with the virus, we are able to start them on antiretroviral therapy within a few hours, and by and large, those people don't end up getting infected. And so giving antiretrovirals really early, like within a few hours after infection, can actually block that infection from occurring. The problem, of course, is that in most types of HIV spread, you don't know an hour later. You can't end up with AZT an hour afterwards. You're not going to go to the doctor and be like, hey, doc, guess what just happened to me? And so we don't have that timing ability to make this happen. We've known for a long time about this idea of post-exposure prophylaxis. That's what we do with the lab workers. As a result, there is another thing that has been developed called pre-exposure prophylaxis, or PrEP. Pre-exposure prophylaxis, so here you can see at the top, ideally you start taking the drugs as soon as possible after having been exposed, within 72 hours. That's what happens when you're a lab worker and you have post-exposure prophylaxis. We have now realized that perhaps we can give some of these antiretrovirals to people who are at high risk of HIV to potentially block them from getting infected. Basically, the idea is to have antiretrovirals in your system at all times to stop transmission from happening. This is called PrEP. The most commonly used drug for PrEP right now is Truvada, which is shown here. PrEP does seem to be very effective, with the caveat that, again, the patient must be HIV negative before starting and must adhere to the regimen. You can't sort of skip a day. You can't only take PrEP on the days that you think you're going to be risky. You have to be taking it very regularly. That was a huge concern when PrEP was first rolled out. And this level of antiretrovirals in your system before infection does seem to block infection. The worry for a long time was that this was going to drive mutation, and that basically we would, by having this drug in the public, drive all the virus in the world to be resistant to this drug, and we'd basically just make one of our drugs useless. That doesn't seem to happen if it's in people's bodies before they even get infected. If the virus has a time to get a foothold before the drug is around, then it can mutate around it. If the drug is there at a good concentration before the virus, then it's going to be okay. We dose this drug at a lower level that doesn't have the side effects that we worry about with others. Yep.
Would you consider that high risk to get
I'm laughing because this was a very hot topic of discussion in my DSEM one day earlier this semester. The way that came up for discussion in our DSEM is that Truvada is now marketed on TV. There are commercials you can see for Truvada right now. You can Google Truvada commercial and you will see it. And they sort of have representative people from each of the high-risk groups. And my DSEM thought that was terrible and wanted to say that everyone can be infected and we shouldn't. They thought it was a very stigmatizing commercial. They had some real problems with the commercial. But gay men are the group that are most frequently prescribed Truvada. They talk a little bit in that commercial, at least, about transgender individuals and African Americans, but you could imagine a lot of different potential high-risk groups. It kind of depends on you and your discussion with your physician. The only place where I've seen it prescribed regularly is in gay men. But you could imagine, depending on a discussion with a physician, other decisions being made. Yes?
So would someone still take this drug if their partner was undetectable?
Oftentimes, yes. But that's a decision that they have to make. I can't make that for them. Yeah.
Why do they require prescription?
Because you need the requirement is to take this drug regularly and at a very specific set of intervals. And so the idea of having a prescription is that then you're going to also have a conversation with your doctor about that need for taking it at the right times and things like that. The biggest worry that was in the field when Truvada was started was that it was going to become a club drug. That people were just going to take Truvada when they go out clubbing and otherwise never take it. And so the idea is if it was over the counter, then there would be much more risk of people taking it sporadically, which would be the situation where we could really see the mutation problem. Yeah, Molly?
How expensive is this in comparison to other areas?
I believe it is cheaper. I am not sure. Yeah, neat.
So if a person acquired it from somebody who was already on anti-retrovirals and their strain mutated around it, would PrEP be ineffective on that person?
It would be ineffective if you happen to acquire HIV from someone who had a virus that was resistant to this drug, yes. And that's one of the reasons why we want to try to limit resistance to this drug. Okay. The other thing that I always want to talk about when I talk about HIV therapies is this. So this is from World AIDS Day 2012. World AIDS Day 2018 was this past weekend. So if you want to look at any news articles about AIDS, they usually are all timed for World AIDS Day, so you can find a lot of things that came out this weekend. But this lists the number of people who were infected and the number of people who had died as of World AIDS Day 2012, and of course it lists at the bottom, cured, 1. And so we also need to talk a little bit about this case, what happened with this case, and what's going on there. The details of this case are quite famous. So there was a patient in Berlin who was really, really, really unlucky. This patient had both HIV and leukemia. as a result of him having HIV and leukemia, there's a whole book called Cured that goes through this whole story in like total detail if you want to know a lot more detail about it. Because of his HIV, because of his leukemia, this patient had to undergo radiation and chemo and get a bone marrow transplant to replace his immune system, which was destroyed by the radiation and chemo. And this was all part of getting rid of the leukemia. But his doctor thought about it, and his doctor gave him a bone marrow transplant of bone marrow that was missing, that had the CR5 delta 32 mutation. So he received bone marrow that was homozygous for this mutation when he received that bone marrow transplant. And the idea was, okay, well, we're going to irradiate his immune system. We're going to get rid of his immune system. That some people will say that would get rid of all the infected cells. Others will say no, his latent reservoir is still going to be there. And then we'll just fill the rest of his immune system with cells that can't be infected. So the virus that comes out of the latent reservoir can never infect any more cells. In 2006, he had this bone marrow transplant. In 2009, he was reported as seronegative without HIV. So that means that his new immune system had never made antibodies to HIV, so the virus seems to have been under control. They can't detect HIV in his system. I was in a lab that received this paper earlier than this for review. I think we might have gotten it in like 2007 or something, and we laughed when we saw it because we said this, we'll say it was like 3 months or something, and we're like, that's nice, wait longer. It's going to take the virus longer to come out of the latent reservoir, but it's going to come out. Like, wait longer, and then we'll care. So in 2007, after 3 years, people started to actually take this seriously when the virus had not come back. And doctors started using the word cure in 2011 when the patient remained seronegative, did not have any ARTs, and his virus still seemed completely gone. He is one of, I would say, there are 3 groups of patients who are very frequently studied in HIV. He gets to be the group himself, I guess, one of the 3 groups. He is very, very, very frequently tested with some of our most sensitive HIV tests now. The virus does not seem to have come back, but what we would expect if the virus were going to come back is we would expect something that looks like this. The dotted line is the limit of detection. So his virus is like really low, it tries to come back, gets up to a certain level, and the immune system shuts it down. And then it's low and up, and the immune system shuts it down. And so we would expect he would have these like little blips of positivity, and then the immune system would shut it back down, and eventually the virus would mutate out of control. He does test positive every so often. We don't know if that is because we are basically using him to use our most sensitive tests. So we're like coming up with newer tests that bring this limit of detection down, 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 down. And sometimes as you make new tests, you get false positives. So we don't know, are those tests false positives because we're designing these super new fancy tests? Or are those tests little blips of the virus coming back that might be expected if it was trying to come out of the latent reservoir? Nobody knows the answer, but it has been 12 years now. He is doing well, and there are other people that have been treated with this same thing and are also doing very well. So as you hear about this, some of you have heard about this from me before, so you know that I can get a little yelly on this one. Do you think that this therapy is going to be a way that we can cure all of the 35 million, or now 36.9 million, infected people in the world? See lots of head shakes. What do we think? Yeah, Emilio. You're going to need a lot of bone marrow that's homozygous for that mutation. Any issues with that? Yeah.
People hate giving bone marrow.
People aren't going to like being bone marrow donors. Okay. Anything else? Yeah, Malik.
The frequency of that mutation?
It's pretty rare. Anything else? There's another big one. Yeah.
Anyone wants to go through chemo?
So you'd have to basically take all 36.9 million people and put them through chemo and radiation. And if you think about doing that around the world, you know, in areas where malaria is present and areas where other diseases are present, it's going to be really hard to treat people where there aren't necessarily doctors and hospitals and great healthcare systems to give everybody radiation and chemo. What about a transplantation issue? Because there's a big transplantation issue here too. Remember how we said the bone? Yeah, Lena.
Like the MHC match?
Yeah. This bone marrow, this mutation is found pretty rarely, and it seems to be very concentrated in one group of people based on sort of where it genetically started in genetic drift. How are you going to get MHC matches with 36.9 million people who are quite diverse from that group of people? So even if you could somehow like convince them to give you all their bone marrow, would it actually be a match for transplant? This is one of the reasons why some people talk about doing things like taking a patient, removing some of their stem cells, and doing CRISPR to get rid of CCR5, or making specifically CCR5 deficient bone marrow for transplant, not making whole babies that have undescribed mutations. And so those types of things might be useful ways of thinking about using CRISPR. But this is probably, and again, you can think about how much that would cost and whether 37 million people in the world could afford it. Those types of scenarios, to me, are not going to really bring down this pandemic too much. It's awesome for this guy and for the other patients who are having it. I'm so happy for them. But I don't know that this is going to have big picture implications on the size of this epidemic. As a result, we can also think about a different way to intervene with HIV. And here we're going to think about using vaccines instead of using therapies. Here what we want to do is we want to prevent infection when a person who is not yet infected instead of curing a person who is already infected. And I will point out there are some ideas for things called therapeutic vaccines where you give a vaccine to someone who's already infected and that vaccine works as a cure. But I'm just going to sort of stick to, just to keep things clean, I'm going to talk about classic vaccines where we're doing prevention here. We talked about the different types of vaccines that you have received in terms of live attenuated, inactivated, toxoids, subunits, and conjugates last time, or last week, once upon a time. AIDS was first observed in the United States in 1981, and the virus was isolated in 1984. Also in 1984, the Secretary of Health and Human Services thought that we would probably have an AIDS vaccine by 1986. In 1997, President Clinton made this huge initiative to get an AIDS vaccine by 2007. And on December 1st, 2012, Ban Ki-moon, the UN Secretary General, says 0 new infections, 0 discrimination, and 0 AIDS-related deaths by 2015 are achievable. As a person who has been in the AIDS vaccine field for a while, I look at these things and say, thanks guys, no pressure. So people have thought that this was going to just be pretty straightforward. Unfortunately, the mutation rate and the variation in this virus have made it very challenging and challenge most of our different approaches to vaccine development. One approach or one issue that I will just generally point out, I'm not going to answer it, one can argue about this quite a bit, is that many of the vaccine trials and many of the experiments that are done right now are funded by the US National Institutes of Health and the US government. And we have to answer this question. We have this very diverse virus that exists in somewhat different genetic forms in different locations. It may not be possible to make a vaccine that prevents against all forms. If not, which forms should we do first in our experiments? And there are some people who say, well, the US government is funding it, you should do the ones that are in the US. And some people will say, no, it's really bad in certain parts of sub-Saharan Africa. We should pick the one they have in sub-Saharan Africa. And there are lots of reasons for support of different opinions on that matter. And so this is sort of a caveat to many of the ideas that are out there. The first group of vaccines for HIV were live attenuated vaccines that were first described. And the first set of live attenuated vaccines that were described involved a virus that was missing one of its 9 genes, a gene called NEF. I'm showing you data from a monkey study with a virus that had a deletion in NEF to talk about how that live attenuated vaccine would work. Weirdly enough, around the time that this happened, there was a cohort of patients found in Perth, Australia, who had a very similar mutation to this. So here you can see there are 4 monkeys who got no vaccine. It says none. And 4 monkeys who got the virus without NEF. And then they all got challenged with some type of SIV. And this tells you about how much virus they have. And to me, the most obvious part of this is if you look at the 4 monkeys who got no vaccine, at some point during their the study, they got dead. Dead, 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 dead. Whereas all of the monkeys who got the vaccine lived. And many of these patients who had the Delta NEF virus in Australia seemed to do pretty well. So people were like, yeah, this seems like a good plan. We think we got it. And then they continued the study further on. At the very top of this figure is the nucleic acid sequence and the amino acid sequence of NEF. Right here is the deletion that was made in NEF. So there were 12 base pairs that were deleted out of NEF, which removed, actually that's not, yeah, 12, that's right, 4 amino acids. And when they looked at the monkeys 2 weeks after infection, after this, you could see that that remained. Well, as they looked at the monkeys further out, this virus actually acquired insertions and rebuilt NEF. So there were single nucleotide insertions at all of those places. First, they made different amino acids, and then they were changing to make amino acids that were more like the amino acids that were actually deleted. So from these data, and in fact, all of those patients in Australia ended up getting AIDS. It was just with slightly slower progression than normal. So based on these types of data, live attenuated, probably not going to cut it. There are some people who still are working on live attenuated vaccines. I'm not a huge fan. And the other reason why the idea of live attenuated may not seem like a particularly great idea is this notion of superinfection that I told you about before. So a person who is infected, like the red lady, can become infected with HIV of a different strain and allow for recombination to happen. And so if being infected with actual HIV doesn't protect you, why would we necessarily think that being infected with this mutant form of HIV would protect you? People then went on to try to do another type of vaccine trial. This study was completed in 2002, and this is a subunit vaccine. So here, a specific GP120 protein was made. GP120 is, again, that surface protein where antibodies are directed against the virus. So this was made in the lab, and patients were injected with multiple injections of this protein. They made clade B and clade E versions of GP120. This was done because they were doing the trial partially in a trial site in Thailand, and this was the predominant virus in Thailand, was clade E. And people had to get 7 shots. So it's not just like 1 shot and then a booster later. They got 1 shot and then 6 boosters. And so I told you about the fact that you have received subunit vaccines and they have been successful before. The The top of this involves the data from this trial. So in the placebo group, 1,273 people got placebo and 105 of them got infected. And in the vaccine group, 1,273 people got vaccine and 106 of them were infected. Or as the newspapers say, it failed or was totally ineffective. So this also was not particularly effective. And this brings us back to what I was saying when I talked about the vaccines lecture, which is that subunit vaccines have a particular problem. This problem is also often seen by inactivated vaccines. These viral proteins are not going to replicate or infect, and so whatever dose you give is not going to amplify over time like a live attenuated. Instead, you're going to have to give separate doses. That's why these patients had to get 7 doses of this vaccine. But there's also no PRR stimulation or other way to help out the immune response to start to induce additional responses. And so these types of vaccines are often coupled with things called adjuvants. And I wrote a paper this summer that talked a lot about adjuvant issues, and when I was giving it to a variety of people to proofread, one of the most common comments I got back is, everybody doesn't know what the word adjuvant means. I guess I was just assuming it, so I'll define the word adjuvant for you since I learned this summer everyone doesn't know what it means. So adjuvants are basically substances that you put in the vaccine in order to enhance or add to that vaccine's efficacy. They're going to help produce a more robust adaptive immune response, ideally with less antigen. There are a few different ways that they can work. One of them, as it says at the top, is about stimulating early processes in the immune response, particularly elements of the inflammatory response. And so this is sort of like the idea when we talked about linked recognition and the antibodies way back when, of adding CFA, that thing with the microbial components, to try to make sure that T cells are getting signal 2 to get turned on. And oftentimes when I think about adjuvants, that is the mechanism by which I think about them. They also can have ways that they work more structurally, where they localize the antigen to a site of infection, or whether they protect the antigen from degradation. So we can see sort of other types of effects. If you are doing a study in animals, you've got lots of choices for adjuvants. Your life is great. You could use CFA, complete Freund's adjuvant, which has mycobacteria in it. Unfortunately, there are very few adjuvants that are actually approved for use in humans. The most famous one is called alum, which is an aluminum hydroxide salt. It's not super effective. There's also one called monophospholipid A, that is used somewhat more and more. There are a few others that are in development. Most of them are things like TLR agonists or other PRR stimuli, like in the paper that I wrote. There's one MF59 that is approved in Europe and not in the United States. It kind of looked like it was on track to be approved in the United States. And then the Italian flu vaccine had a bunch of side effects one year, and so then it sort of went back on hold a little bit. And so one thing that perhaps we could do is we could repeat that trial with the HIV subunit vaccine along with some adjuvants in order to improve efficacy. One big type of thing that people often talk about as adjuvants, and again, I might be biased because my PhD was on doing this, is the idea of including a cytokine in with the vaccine so that you can push the T-cell response in the direction that you want. And so we can use a lot of our immunology knowledge to help us design good adjuvants. There are also some other types of technologies that are being investigated for vaccines. These are the recombinant vector-based vaccine and the DNA-based vaccine. They both have some similarities. In both cases, we study the pathogen of interest and identify the section that codes for a particular antigen. So here, this red RNA codes for this little red protein. we can then either just put that piece of nucleic acid into a plasmid and inject the plasmid directly, or we can put that piece of nucleic acid into another pathogen that, because it's not a pathogen, into another microbe, which is harmless, and allow that microbe to now make the antigen of interest and inject that as a vaccine. DNA vaccines are still very much in trials. There are none that are done that have been completely approved yet. They do seem to be pretty good in terms of their immune responses. The idea is that basically you actually deliver them intramuscularly, and the idea is that you're getting the muscle cell to start making the antigen and presenting directly on class 1 to start stimulating T cells. The live vector vaccine has been in a number of different clinical tests. The great thing here is that it replicates, so you get amplification, and you get PRR stimulation because you have an actual pathogen. And so it sort of adjuvants itself, but it's potentially safe. In general, we talk about the fact that these have not really been approved, but in fact, the Ebola vaccine that was used as part of the 2014 Ebola outbreak and that is currently being delivered in the DRC is a recombinant vector-based vaccine where an Ebola antigen is put in another pathogen that makes that pathogen have this one Ebola antigen and induces antibodies against Ebola. That type of vaccine is known as a recombinant vector-based vaccine. There is one recombinant vector-based vaccine that has been tried for HIV. I have a lot of feelings on this one too. A lot of feelings on this one because this one was actually a human trial based on a trial that my PhD lab did in monkeys. And I'm going to try to say it in the nice way now. So it was done, the trial is known as the STEP trial. It was done by Merck. And they had a virus known as an adenovirus that encoded antigens from SIV. And patients were given this virus at day 1, week 4, and week 26. This was not exactly the way that the monkey trial had been structured for reasons. One problem with this vector-based vaccine, just like any vector-based vaccine, is that you have to think a little bit about the specific vector that you're using. In this case, we're using a specific type of adenovirus. And if you think about it, you might be like, well, what about people who already had adenovirus infections, who had adenovirus when they were a kid, who have an immune response to adenovirus? They might have antibodies that actually kill the vaccine before we have time for it to work as a vaccine and induce an immune response. This is a common problem when we think about vector-based vaccines, and so we have to think carefully about our vector, both that it's not pathogenic, but also about the levels of pre-existing immunity. As a result, this trial had the data segregated based on how much antibody the patients had against adenovirus before the trial started. And the idea was, well, maybe people who had a lot of antibody are probably going to have it not work, and the people who didn't have any antibody will have it work. So here at the top, you can see people who didn't have too much antibody. You can see that between vaccine and placebo, no big difference. But they found that in patients who had a very high amount of antibodies, it seemed as though those with the vaccine had an enhanced rate of infection. The vaccine made you more likely to get infected instead of less likely to get infected. This led to kind of a shutdown in the field of vaccine research for a while. There are lots of questions about how this may have made people, may or may not have made people more vulnerable to HIV. Some people think this was about changing T-cell activation to make more target cells. Other people say that if you actually rerun the statistics, when they made these two groups, when they made the placebo and the vaccine group, they randomized those groups for things that they knew were important factors in HIV transmission at the beginning of the trial. Turns out they didn't know at the beginning of the trial that circumcision was an important risk factor for HIV transmission. And if you go back and look now at the percent of circumcised individuals between the two groups, they are very much not equal. And it's entirely possible that the entire effect that was seen was based on circumcision and randomization error. And if you redo some of the stats, you can also see that. So it's not really clear what's going on. I mentioned before that there are 3 groups of patients that are some of the most studied in the HIV field. These people who got sick but got this vaccine are one of the other groups that everybody studies all the time to see what's going on. So it's not really clear. There have been a few more studies that have been completed since then. Another one was also a vector-based vaccine that used now a vector called canarypox. And it did show 30% reduction in HIV acquisition. And there are even more vaccines that are all out there currently in trials. By and large, they are all recombinant vector-based vaccines. And that is the place where most of HIV vaccinology is going forward. I'm going to skip, but those are such good slides. All right, I'm doing them anyway, and I'm going to be behind, and I got to figure out how I'm going to get these other ones too. Because everything I got left is awesome. One of the things that people also often think about when they try to think about dealing with HIV is about the neutralizing antibodies. So we see that neutralizing antibodies can be really useful in protection from HIV. And we can even find that there are some epitopes where we can have antibodies, and if those antibodies are made in a patient, that patient will make antibodies that can neutralize the virus, and that can actually neutralize the virus across multiple different virus groups. They're called broadly neutralizing antibodies, or BNAbs. We know a lot of the different epitopes against which these antibodies are directed. We find them in some patients, but not all. It seems as though one problem is that glycosylation of GP120 and GP41 often blocks some of the antibody responses. There's one that's listed here as 4E10. I believe it's 4E10. There's one that's really sort of close to the membrane, and it turns out that it actually makes some contacts with the membrane as well as with the spike, and it's cross-reactive with a heart lipid. And so all the B cells that make it are deleted. Those B cells are found in some lupus patients, and it seems as though there is a reduction in HIV levels in lupus patients, as though there's some cross-reactivity. And so one reason why some patients might not make some of these neutralizing antibodies is because they might also be self-reactive because of that lipid binding. But people have spent a lot of time trying to characterize these neutralizing antibodies, because if we knew what these antibodies were like, we could potentially try to do immunizations to make them emerge. So what people have actually done is there were a cohort of patients who were studied for a huge amount of time. Their antibodies were taken and their virus was taken and sequenced. And those who made the neutralizing antibodies were then examined to see how did their antibodies evolve over time and how did their virus evolve over time, with the idea of designing a series of antigens that match this evolution to push the antibodies to become those broadly neutralizing antibodies. These antibodies seem to have particularly long CDR3 regions and to have undergone extensive somatic hypermutation such that designing them could be a particular challenge. One of the antibodies that was described as the end of all this was one called VRC01. And the paper that we're reading on Friday is an alternative approach to getting VRC01 in patients. I will see you guys on Wednesday when we're going to talk about cancer, and sometime I got to finish this other vaccine stuff too. I really like vaccines. I know you're shocked. See you on Wednesday.